students, welcome back to the online class. Yesterday we have seen about the structure of the nephron. Today let us move to the next part, that is the mechanism of urine formation. Okay, so try to recall the things yesterday what we have seen in the structure of nephron. We have told that there are two important structures. One is the Malpighian body and the second one is the renal tubule. Okay, right. Malpighian body is made up of two things. One is Bowman's capsule and the other one is called glomerulus. So what is this Bowman's cup or Bowman's capsule? This is the part. So you can see this orange colored structure. All this is the total structure of the nephron, and this part. Okay, so this part here, the round circular part here. Okay, this is nothing but the Bowman's cup, a cup-like structure. So in which a network of blood vessels are present, which is called as glomerulus. So because of this glomerulus only, so the filtration process will take place, and this is what we call it as filtration unit, right? So this was the first part. Second part is nothing but the remaining tubular portion. So if you see this highly long lengthy tube like this is the renal tube, we call it as. In this again there are three parts. First part is called PCT, proximal convoluted tubule. Yesterday in detail we have seen that. Okay. Second part here you can see this U-shaped structure here, which is called as the loop of Henle. Okay. And lastly from here onwards up to this part we have got DCT, distal convoluted tubule. Right. So these are the three important parts of the second portion, that is the renal tubule. Right. Now today let us see about how the mechanism of the urine formation takes place, and this is studied under 4 stages. So what are those 4 stages? Stage number 1. So let us see here. So this is called as the glomerular filtration. First stage is called as the glomerular filtration. So where does this take place? It takes place in the glomerulus. So this is the glomerulus. We have put a network of blood vessels. How is this formed? It is formed because of the incoming artery. Which is the incoming artery? Afferent arteriole. It is going inside and it comes out as efferent arteriole, outgoing. Okay, so coming out blood vessel is called efferent arteriole, and this efferent arteriole itself forms a network of blood vessels, which is called as peritubular capillaries or capillary network, we call it as. Okay, which is useful in the reabsorption second stage, right? So the filtration which is occurring in the glomerulus, so that is called as the glomerular filtration, or it is called pressure filtration, or it is called ultrafiltration. Okay, so what happens during this? So because of the narrowness of the efferent arteriole, high pressure is developed at this point. Okay, and the things are released, the waste metals are released into the Bowman's cup. So the green color arrow marks we can show it, we can see here, moving into this cup, bottom portion, lower portion of the Bowman's cup. So what are entering? So waste molecules, nutrient molecules and water, they are filtered and sent into the Bowman's cup. From here slowly what happens? Now the waste metals start moving into the first part of the renal tubule, which is called as the PCT. So in this way, this flow of direction you can see here, arrow marks how the urine will be moving here, and the first formed liquid which is there, okay, nephric filtrate we call it as. So that is called as the primary urine. So these are the directions to show how the urine travels in the entire tube here. So the white color arrow marks you can see here. So how the urine will be moving and finally into the DCT to the collecting tube like this. Okay. So the filtration occurring here, it is the first stage, it is called as the glomerular filtration. It is also called pressure filtration. Why this pressure is developed? Because of the narrowness. So this part, so a very narrow tube is present. So because of that, high pressure is developed, then the filtration takes place. It is also called as the ultrafiltration, and the substances like waste molecules, nutrient molecules, water, all these are sent into this lower part of the Bowman's cup, or in other words we can say first part of the renal tubule, which is called PCT, proximal convoluted tubule. Okay, now let us move to the second tubular reabsorption. Okay, third stage is the mechanism of the urine formation, that is tubular secretion, and fourth stage formation of hypertonic urine. Okay, what happens in the tubular reabsorption? Okay, coming to the second stage now, tubular reabsorption. So what do you mean by reabsorption? So that is picking out the useful materials to the body. So whatever are important for the body, it's not that the urine which is formed, primary urine itself is eliminated outside. There are many things which are present in the urine. Okay, already we have seen in the in the beginning of the lesson we have seen blood analysis report and urine analysis report. Okay, the different type of 13 to 14 type of substances are present. So some of them are toxic which are to be eliminated, and the things which are useful are to be sent back again into our blood. So who will do this work? So it is done under the second process, that is the tubular reabsorption. So this urine which is formed in the beginning, which travels from the Bowman's cup into the PCT, proximal convoluted tubule. Okay, so there the urine which is formed, it is called as the primary urine. We call it as primary urine, and this is almost equivalent to the blood in the chemical composition. Whatever substances are generally present in our blood, all such type of things are present in the primary urine here. So in that again, whatever the useful substances are present, so they are reabsorbed. Okay, taking it back into the body is nothing but what we call it as reabsorption. Then who will do this? So in the renal tubule, the anterior part is there, PCT, proximal convoluted tubule is there. Okay, so in that region, whenever the urine is passing, so primary urine is passing, then the reabsorption process will take place. So which type of the blood vessels are important here? We have told yesterday that the total renal tubule is totally packed up with a network of blood vessels. So what are they called as? Peritubular capillaries. So they help in the process of reabsorption of the components of the primary urine. Okay, whatever the important components are there useful to the body, so they are taken back again. Before they are eliminated, they are taken back. Okay, what type of things? Like we have got glucose C6, H12O6. Okay, amino acids are there, that is the end products of the proteins. Then we have got vitamin C. So in the beginning we have seen in the first chapter, that is the nutrition, there are two types of the vitamins. Okay, water soluble and fat soluble. So one of the example for the water soluble is the vitamin C here. So water soluble vitamin C, what you call vitamin here. Then next salts like we have got potassium, calcium, sodium, and next we have got chlorides and 75% of water. All these things are what? They are reabsorbed. Most important, they cannot be eliminated directly outside. So once again, glucose, amino acids, vitamin C, potassium, calcium, sodium, chlorides, and 75% of water. So this is all happens in the PCT. So by the peritubular capillaries which are surrounding the renal tubule. Okay. Now moving to the third stage of the mechanism of urine formation, tubular secretion. And this particular tubular secretion occurs in which area? DCT, distal convoluted tubule, the posterior part of the renal tubule, or in other words, the third part of the renal tubule is called as distal convoluted tubule. So we have discussed about this in detail in the structure of nephron in the earlier class. Okay, DCT, distal convoluted tubule. So this area, what happens? Tubular secretion takes place. So what type of things are happening here? So whatever the extra salts are present, ions are present, so they are sent into the urinary tubule here, and the removal of the waste products. Here we have seen useful substances are taken back. Now here we have to see the removal of the waste products. Removal of waste products from the blood. Okay, so what type of waste products are present? In the beginning we have told the end products of different type of the metabolic activities in our body. Okay, so like we have got urea, uric acid, then we have got creatinine. Okay, I suppose you remember about this creatinine. This is a good indication. So by the increasing level of it in the blood, we can imagine what is the functioning of our kidneys. So if it exceeds more than 2, then it is a very danger levels we have got. Okay, creatinine. So this creatinine is also left in the primary urine, which is now passing through the DCT, distal convoluted tubule. Okay, so urea, uric acid, creatinine, then salts like potassium. Okay, potassium, then next sodium ions and hydrogen ions. All such type of things which are now considered to be as waste, so they are to be removed out. So that is what we call it as a tubular secretion. And once this thing happens in the DCT, distal convoluted tubule, okay, then next goes on with the fourth type of the fourth stage of the mechanism of urine formation, that is formation of hypertonic urine. Now all the useful things are taken during reabsorption, all the waste things are now removed during secretion. Now it's the time, okay, formation of the hypertonic urine. And as long as the urine is moving in the distal convoluted tubule, so here maintenance of the proper pH, okay, concentration of urine will take place during this process called as tubular secretion. Okay, now let us move to the next part, hypertonic urine. So formation of the hypertonic urine. So here 75% of the nephric filtrate. So what do you mean by nephric filtrate? The liquid which is formed after the filtration done by the nephrons. Okay, once again, the liquid which is formed after the filtration done by the structural functional units of the kidneys called as nephrons. 75% is nephric filtrate. Okay, reabsorption takes place in the proximal convoluted tubule. So already we have told reabsorption, tubular reabsorption is taking place in the PCT. So maximum, okay, 75% reabsorption is happening in the proximal convoluted tubule, first part of the renal tubule, or in other words, second part of the structure of nephron. First part is Malpighian body, second part is renal tubule. In the renal tubule, first part is PCT, proximal convoluted tubule. You can see in the figure which is PCT there. Okay, now after that, so 10% of the water, okay, which passes through the process. What is this process? Osmosis. So already we have studied in the earlier class, movement of the ion from low concentration to high concentration, that is called as osmosis. So 10% of the water passes through the osmosis in the area of Henry's loop. So what do you mean by this Henry's loop? In the structure of nephron we have seen the middle portion of the renal tubule. You can see a U-shaped tube like this. Okay, U-shaped, capital letter U, U for umbrella. So this shaped tube is called as what? Loop of Henry, or Henry's loop we call. So 10% of the water passes through the osmosis in the middle region of the renal tubule, which is called Henry's loop. Right? The next concentration of the urine takes place in the collecting tube. So here collecting tube. So we have told the last part after the DCT, the next part which moves into the pelvis region is the collecting tube. It collects the waste materials. Okay, in the presence of a most important hormone which is called as the vasopressin. Okay, very important hormone. This is vasopressin, and this is secreted when the concentration of the urine is passed out. Okay, and this particular vasopressin hormone is very much important, and its absence, the absence of the vasopressin, okay, so it produces dilute urine, more occurrence of the urine, liquid, okay, more liquidity will be there, that is the dilution will be there, dilute urine is passed because of the absence of the vasopressin, and this leads to one condition or a disease we can call it as, which is called as the diabetes insipidus. Okay, so the produces this vasopressin, absence of the vasopressin produces the dilute urine, and it maintains, vasopressin maintains the osmotic concentration of the body fluids. So whatever the different type of the body fluids are, the liquids which are moving in the body, so that concentration is maintained by this hormone called as the vasopressin. So deficiency of this causes excessive, repeated and dilute urination. So passing of the urine is called urination, and its absence leads to a diseased condition which is called as diabetes insipidus. Okay, so this diabetes is caused because of the absence of the hormone called as vasopressin. So it helps in controlling the flow of the urine from our body, and the people those who have got absence of this vasopressin, or in whose body this required quantity of vasopressin is not produced, near about 13 to 15 liters of urine is passed per day in 24 hours. Okay, so this is the very large quantity of the urine passed. That means for every 10 to 15 minutes the person has to go for the urination process. Okay, so it helps in controlling the flow of the urine in our body. Okay, so vasopressin plays an important role. So leads to its absence leads to a condition diabetes insipidus. There is one more condition which is called as diabetes mellitus. Diabetes mellitus. Okay, and about this you are going to study in the next coming chapters, where the sugar levels will be more recorded in the blood, and this is due to the absence of or insufficient production of one hormone which is called as insulin hormone, and by that when we take this insulin hormone, then it controls the this particular disease condition called as diabetes mellitus. Okay, diabetes mellitus, and this is diabetes insipidus. Right?